So now actually we get to the big question. What happens when we mess around with the first receptor? And the thing is, for all these different senses, they all look like G proteins at a certain level. 3 of our 5 senses are G proteins. And I'm just not sure about touch and what's the other one? Light, smell, taste, touch, hearing. Touch and hearing are a little bit different. They're different kinds of things. But these are things with clear first messengers. And we use that. So that's what's going on when you do this. Can you see that this person, she's looking at Ayers rock, and you can see it reflected in the eye. Of course, some of this light is going through and hitting the back of the eye, being detected by the eye itself. And it turns out when you look and see what's actually detecting the photons, what is changing its shape when light hits it, it's another G protein. So it's a G protein coupled receptor. If you look at this, same 7 helix thing, and even a small molecule that's sort of bound deep into the receptor, just like we see the binding sites for it. Zach?
Is the smallest response to light in G proteins 1 G protein?
Mm-hmm. 1 photon can activate a human eye nerve response, which means 1 photon can activate this whole thing. Yeah. And basically, you don't need to be any better than that. That's optimal. You don't get any survival advantage from being able to detect half a photon. And there's a paper about that, by the way. If you're interested in more details on that, I can look that up.
Is there a variation of G proteins that respond based on color, or is that not?
Yes, and that's coming up in just a couple slides. So to sense light of any wavelength, you have a thing that looks like this. And again, we have red protein, small molecule, green protein. But the small molecule is doing something slightly different here. This is rhodopsin, and the small molecule is retinal. Here's transducin, which is a G protein. And you can see again, I believe on this one, this is one that where the beta and the gamma are the ones that might be leaving, like the other one. Actually, I'm not sure. I won't say anything. We'll let the molecular biologist tell us which way it goes, okay? The main thing is, this looks like everything else I showed you. And it's at the heart of this whole signaling system. Any biologist want to guess what species this is? Definitely looks insect-like, right? I don't remember exactly, so I'll let it tell me. It's a dialonglegs, so very close. It's a dialonglegs eye. I'm not sure why people would study that. Maybe they're easier to look at in some ways. But there's green lenses. They focus light on the cells right here, and these cells are connected to nerves. So this goes back to the neuron lecture. We have neuron signaling going on here, but how do we get the signaling started? Here's the fly eye, for those of you who want Drosophila. The retina is in brown, the photoaxons are in blue, and the brain neurons are in green. So light comes in here, hits this, and it travels through. And so our eye looks just like this from this level of detail. Does anybody know how our eye is weird? This makes sense, right? This is the way you'd arrange an eye. But our eye is not actually arranged by that. I see two hands sort of going up. Who wants to take it? Or are you just scratching? Okay, so Brian, what do you think? What's backwards? Mm-hmm. That's flipped around. So really weird, good evidence for evolution. And yet, remember what I said? A single photon can still activate it. So it's messed up, but it doesn't matter. It's all a question of nature is not perfectionistic. And so don't feel like you have to be either. Zach?
Is that a consequence of the whites of the eyes?
Oh, biology. I'll plead biology. It's a consequence of, I think you can look back at it evolutionarily and come up with some kind of trajectory. But it's about how cells are arranged. And I'm happy to talk about how proteins work, which, by the way, is more efficient because the proteins can respond to a single photon. So I get to talk about the good part. Okay? This is the arrangement. And you actually see, here's the rods and cones. I literally thought Linenger had a typo when I first showed this until the biologist set me straight that Linenger was right. You have the rods and cones down here. Those are the photoreceptors. And then you have the ganglion cells and other cells that are on top of them. The light's coming in like this. Not the best arrangement, but it works. We survive. We pass on our genes. Life is good. Okay? So this is a correct way to show it. Light comes in like this, and it hits the rods and cones. And then you have a bunch of neurons. Once you get to this part, it's all neuron signaling. So our question really is, what's going on in the rods and cones? How does light get turned into a voltage? So if you look at the rods and cones, they have weird shapes, and they have a bunch of disks. The disks are made of membrane. So that's our first clue that what's important in them is going to be a membrane protein. Well, what are Gproteincoupled receptors? Membrane proteins. And these are stuffed full with those and with retinal. Cones have the same thing. Cones look the same. They have a different shape, but they have the same kind of disks. And in fact, rods and cones are very similar. And as far as we're talking about starting the signal, they work the same. Lots of membranes. And there's also ion channels. And so there's going to be polarization due to ion channels. So it's like a neuron, but there's one important part of it that's flipped around from regular neurons. So the question is, how do you start? How do you send a single photon into the cell? And how in the world does that get amplified into enough ion channels opening that you cause a voltage big enough to send a signal? Here's a mouse eye. We're going through all the species. This is a nerve fiber layer. And by the way, extremely important sense, extreme amount of signaling going on here. The optic nerve, is it the most active nerve in the body, the optic nerve? I know there's things about like keeping that nerve active results in a lot of residual damage, and frequently blindness can result from optic nerve trouble because it's like the superhighway of nerves. Is that right? Yeah. Yeah, it's extremely dense. And remember that all of those are running off of ATP. And so if you have this dense network of ATP burning stuff, oxygen burning stuff, you're going to have a lot of damage from the reactive chemistry going where it shouldn't. So it seems like this is going to be, but clearly it's important enough to the organism that you invest a huge amount into this. So the way that it works, this is what's backwards. It works by changing ion channels. Before we talked about how neurons work by opening ion channels, right? Sodium, then potassium opens slower. This is the reverse. It actually works by an ion channel closing. When light comes in, an ion channel closes, sodium and calcium can't get in anymore. Remember, you've got the ATPase constantly pumping, trying to keep the signal going. But here's like, instead of opening a door, they leave the door open. When the photon hits, they close the door. And then the ATPase is still working away, and you end up with hyperpolarization rather than depolarization. Is that the right word? Yes. Because you end up with a larger voltage when the signal is sent. Most neurons work the opposite way, but most neurons don't have the same job that this does. It turns out that this hyperpolarization is actually probably the best way to do it, to respond to a very faint stimulus. So you got the ATPase here, by the way, and remember that it's constantly working. You close the door, it works better. Then the rest of the stuff, so how do you get from a photon coming in to the door closing? Well, you go through a cascade that looks a lot like every other G-protein-coupled receptor. The light hits the rhodopsin, and way back when, at the beginning of biochemistry, we mentioned double bond can flip around, and that can cause the rhodopsin to straighten out, and that will actually push the G protein off, and that will cause it to bind G, and then it will go and do its stuff. Everything else here is exactly the same but flipped on its head, because what you have is you actually have a phosphodiesterase that is turned on. Instead of a cyclase that's turned on, you turn on a phosphodiesterase, and it eats up the cGMP second messenger. When the cGMP second messenger is what's binding to this door and keeping it open. So it's the door stop keeping the door open. You eat it all up, and the door closes, and then you have hyperpolarization. My suspicion is that this is all because this works better with a constantly active cell. Even though it burns more energy, it's not as efficient, but it's more sensitive. The main thing over here, ion channel closing, therefore membrane is hyperpolarized. But don't let that fool you. It's basically the same thing turned on its head, right? Now, turning off the signal is kind of the same thing in reverse. You have a cyclase that will eventually make cGMP that will actually reopen the door and turn off things. You also have an arrestin that will go and it will bind the rhodopsin and it will kick out the retinal. So it can't very well signal if it doesn't have the light binding molecule, the light absorbing molecule. And so you have multiple ways to turn off the signal. But again, when we get down to the bottom part of all of these figures, by the way, that's when you can actually start to skip over stuff. What's most similar is the top part of the figure, and I want you to see, we only have time for the similarities. You can dig into the differences on your own time. Mirko? Yeah. Mm-hmm.
So what I know is that I adapt to different sunlight, but
Mhm.
not doing any I adjust.
Yeah.
What if you actually are sleeping, right? You're not fully done.
Mm-hmm.
You still get light going through. Now, does your eyes feel like continuous amount of energy even when you're sleeping? How does it work? How does it adjust to this?
Yeah, I think there might be some mechanisms like this at a totally different level. Yeah, I think you might have some major things, because it makes sense that you wouldn't keep the optic nerve if it is so costly to keep going. Now, realize when it's active, when you have a lot of light coming in, that is going to still burn more energy than when you have no light coming in, because the hyperpolarization costs more energy than, in a sense, to maintain. You're going up in volts rather than down in volts. But yeah, we can. All I can say is there's lots of ways to turn this off. I think there are some brain level ways to turn off the signal, or at least dampen down the area so that you aren't burning as much. But that would require study of the whole nerve in a whole organism. And again, I'm happy to let the biologists take care of those whole organism type questions. Although, you know, a sleep study, I could go for that, you know. Yeah. And this weekend, hopefully, you'll have a little bit of a chance. Okay. So this is really signal amplification. Believe it or not, we are actually close to the end of the lecture. One photon can excite one rhodopsin, and that can result in all of this polarization. And that leads to the question of, is the eye optimized in terms of its sensitivity? It is not optimized in terms of its anatomy. But we're not made any guarantees to whether anatomy is going to be optimized, honestly. What's important for survival is being able to respond to, at minimum, one photon. And that's the argument. Are we there or not? If we're not there, we're really close. And so, one rhodopsin can do 500 molecules of transducin, 500 activated PDE molecules. These are so much bigger than the other numbers I showed you. That's why I'm saying I think this is more costly, but also produces more signal from a fainter first messenger. And this is just the PDE. Each PDE is an enzyme that and we can calculate how many GMP per second it can eat up. And that's lots of GMP, okay? So we can just say that there's millions of GMP that can be eaten up by one photon. Single photon, thousands of ion channels closed, membrane potential change that we can measure of 1 millivolt. One photon, 1 millivolt. There's a lot of amplification going on there. Thanks to ATP, by the way, we're not breaking the second law of thermodynamics, or the first law, but that we're getting the amplification out because we are constantly burning ATP. So I want to give you one really complex story that's kind of hard to understand, okay? So you can guess whether I'm going to ask a question about this. This connects everything from chapter 12. And it's a story about an anesthetic that lets mice see. An anesthetic. There's a genetic disease that causes blindness. And you can inject a chemical photo switch into the mice, and all of a sudden, they can see. The pupil of the mouse on the right was injected with the chemical, and suddenly the pupil starts contracting. It starts responding to light. And you can see that the nerves are actually responding to the light. This is a very specific genetic condition, but it turns out that this molecule is an anesthetic of a sort. These mice are homozygous for a mutation in the PDE, and it causes the rods and cones to degenerate by 3 months of age. They don't have any rods or cones. That's what makes this more amazing. It's not like we're bringing rods and cones out of the drawers here. Or it's not like they have one protein that's messed up, and we're injecting something that's fitting into that protein and making it have the right shape again. This is an entire type of cell that is gone, okay? So how in the world does this work? Well, this chemical turns neurons into light-sensing neurons. And it's called AAQ. It is a light-activatable potassium channel blocker. Now, if I gave you like 15 minutes to talk this over and figure it out, I think that you could put the pieces together and figure out how it is that a potassium channel blocker could let blind mice see. But we all want to go home. So let's do this. When visible light hits AAQ, this is AAQ. It looks kind of like rhodopsin, actually. It's got a bond in the middle. In this case, it's an NN double bond. When 380 light hits it, it flips around like this. When 500 light hits it, it flips around like that. When 500 light hits it in the visible range, this conformation sticks in the middle of a potassium channel and blocks it. When infrared light hits it, or actually that's UV, sorry. When higher energy light hits it, or when it untwists by itself, then it will leave the channel unblocked. If you give the mice this, it will go to their potassium channels in their eye, and it will sit there and it will block them, and it will change whether it's blocking or not based on the wavelength of light coming in. This is how anesthetics work. If you remember, they will block the different channels, and I believe this one actually is an anesthetic in other cases. They will block ion channels, but most of them are not changed by light. This one being light sensitive and being injected into the mouse eyes is what makes it work. Mirko.
How are they sure that it affects only the eye and the nerves? So both have potassium channel.
Well, they know it's not, they don't have time to grow new rods and cones, so they know that it must be working, and they know that it's specifically, at least its major effect is that it is a potassium channel blocker. So when you see how it works, I think that you'll see that this is the major plausible mechanism for how it can work. Yeah, they don't have any rods and cones, so they would have to build them if they had to build them.
But how do you know that it wouldn't affect the neurons itself?
Oh, they probably do. It's probably, these mice might be like awakenings, you know, like where they're healed and then they get sick again. Is that a spoiler? Anyways, so sorry about that, but it's an old movie. I remember it's back when it's Robin Williams and I forget who else. Anyways, Oliver Sacks, good story. So the main thing is, though, if you block the potassium channel is turning the polarization off. When the nerve is activated, the nerve opens the sodium channels and it gets rid of the positive charge. It hyperpolarizes, and then it depolarizes again by opening these up. But that's blocked, and so the positive charge can't go anywhere. And this is how anesthetics work. They are not, most anesthetics will work this way. They'll block channels. This is just one that it will block the channel of the neuron, but then when light hits it, it unblocks, lets the signal out, and that ends up being enough in that context to send a signal when light hits it. When light hits it and it blocks the channel, it's enough to, and apparently neurons are firing enough at a low enough level, they're just on the verge of firing, that this will work this way. This is complex, but think it through and go through to it. We are at 420 right now, but I've got a little bit left in this part, and then I've got the cancer part to talk about. So like I said before, I'm recording this, but stay for another 10 minutes if you can. Otherwise, watch it online. All right. So cone cells, how do they sense light? They're basically just like rhodopsin, but they have different protein environments that will shift the wavelengths that they absorb. Rhodopsin absorbs right in the middle of the visible range. But it looks like these other ones are just like rhodopsin, but they've been shifted to absorb on one end or the other of the range. And that's what you have in cone cells. So it's not very different. You just have a blue mutated version, a green mutated version, and a red mutated version. Rhodopsin is the most average of these options. And the cone cells are sort of pushed to the edges, and then you get information from integrating the information from each of those cones. Now, we know about this now because we know how this works, and we know that color blindness happens if you don't have one of these opsin genes. Like if you don't have the green one, you can't distinguish green from red. You can still see the light, right? You just can't distinguish it. And that shows you that the different opsins are important for distinguishing. Cone cells are for distinguishing colors, not for necessarily detecting it. You've got tons of cells that can detect light. So Dalton was colorblind, and he didn't know why. They didn't even know that genes were doing anything at that time. His hypothesis was that I think I'm colorblind because I have the wrong color inside my eye. This actually is probably the most parsimonious option available to him. And of course, he wasn't going to cut open his eye, but he said, after my death, I want you to cut open my eye, and I want to know if I'm right. Okay? And the fluid was normal. So his last hypothesis was wrong. I'm sure he's probably okay with that. Okay? But the eyes were saved, because you save everything, right? And in the mid-90s, we got technology to be able to take the DNA and sequence what genes were in those eyes. And it found out from PCR that they could find all the opsins except for the green. And so he did not have a green opsin, and it was, as we thought, the most common kind of color blindness, which is missing that opsin. There's other weird things about light. There's people with, who don't have a lens on their eye, which may have a filtering purpose. There's people without a lens on the eye who report that they can see wavelengths outside of ultraviolet light. And Monet claimed to do this. He actually underwent cataract surgery. I think they took out some of his lens, and he claimed to gain the ability to see UV light. And it caused him to paint everything sort of blue shifted. It's like his world was being lit up by a black light. And if you ever wonder why a dorm room poster, do you guys still do black light posters? You have one person on the dorm who does black light posters. They used to do it in the 70s and 80s, okay? But black light, it looks like a black light poster. It could be because his lens was gone. And then there's another weird thing about color. You're integrating these different signals to result in a single perception of a color. Well, if you look at the way the spectrum works, there are certain colors that we should be able to see, but we can't. If we look at the wavelength is just changing smoothly, but the perception of color changes abruptly. We should be able to see a red-green color or a blue-yellow color, but our brains don't let us interpret those as intermediate colors. It forces us to push them one way or another. So there was an experiment in 1983 when they figured out that we could actually do striped images where you mess with one half of the retinal cells and you basically overload the eye, and then apparently you can see these colors. I don't know what the IRB on an experiment like that would be, but and it's also not my field, but maybe you'll find a psychology professor who will let you try to see the forbidden colors. Sounds like a Harry Potter, bad Harry Potter story, Harry Potter and the Forbidden Colors. Here's something that just happened, and this is one of the reasons why I wanted to do this lecture this year. If it's all about the opsins and it's all about the genes, well, can't we do gene therapy? Now we can. This just came out, that mice have been given a cone opsin and their color blindness has been turned around so that they can now distinguish. I believe it's the cone opsin, so I believe it is, I'm not sure what color it is actually, but it's about seeing some wavelengths they couldn't see before. Gives them the opsin. And it really is a viral gene therapy delivery. This is more like the kind of paper that you can look into if it fits into one of our categories. And they actually inject the virus in, and the virus goes in, and it actually puts the gene into one of the cells. I think it's, I'm not going to go into the details, but basically it's gene therapy and it's working. The main thing about this is what's really working here is doing things simply that they never did in the first place. They moved on to the more complex kinds of ways of doing experiments, and no one ever tried this simple experiment where they just take the opsin and they basically send them into the ganglion cells. I think it's which cells you send them into that's important. If you send them into the other cells, they don't accept the gene. But ganglion cells will work, and they will fix the gene, and they'll start responding to the light. And it's what they're saying, you could have done this 20 years ago. They tried a couple of combinations, different cells, different viruses, and things like that, but they never tried this one until they came about it as a roundabout kind of way through another experiment. So that means that some gene therapy can work, and but it's finding the right combination. There's another, this is a bit more sci-fi, but you have nanoparticles that will glom onto cells. They're just particles of metals that are very small size, but they're brightly colored, they respond to light, they absorb light energy. If you just take these, they will automatically go onto cells, and you put them in mice. They are ones that absorb near infrared light, and then they emit visible light. That's the way that their energy transformation works. If you give these to mice, they'll go to their rod and cone cells, and they'll attach to them, and they will give them the ability, because they convert infrared to visible, then the visible light will be picked up by the cell, and the mouse will respond to infrared colors. So talk about forbidden colors, we have forbidden UV, forbidden IR, and forbidden in-between visible colors. All of this has to do with this one signaling process. And so messing with this process can possibly make really interesting things happen. Okay. So smell and taste, honestly, when I teach this in class, there's part of the test that is about this. For you, you don't really have to worry about it too much, except that you can see the same patterns of colors that we had before. Smell, also G protein coupled. You have the red odorant receptor. You also have some binding proteins out here, by the way, which complicate things. Odorant receptor, you have a G protein. The alpha part of the G protein binds adenylyl cyclase. You have CAMP, second messenger. And then you have some ion channels opening. The right part of this is always going to be a little different, but the left part looks exactly the same as what we've seen before. So smell, G protein coupled receptor. Very similar division. And by the way, this is what Linda Buck won the Nobel Prize for in 2004. And she figured this out. A smell researcher works at a cancer institute for reasons that I had before, and we'll touch upon them in the last part. Taste is also G protein coupled. Red, green, blue. Left side of the figure looks exactly the same. Even CAMP is produced. You even have pKa being involved. So you have a lot of the same things going on, a lot of overlap between these. So we've got a lot of G proteins. We've talked about GS, GI, and transducin. The olfactory ones are called G-olf, and the taste ones are called G-gust, okay? But they're all Gs. That's the main thing. You can find cool things like this. I really like this because it shows the parallels with all the red, green, blue at the top, the second messengers in the middle. You can have different second messengers going up or going down, depending on which way you're doing things. They mostly tend to go up. And then you can have kinases or ion channels opening or closing. But right here, this is sight, taste, smell, and adrenaline, all on the same figure. So you can tie this back to insulin. Glucagon is another molecule that sends a signal that's pretty much opposite to insulin. Did you talk about glucagon in Biochem 2? It turns out that glucagon also works through a GS pathway. It also has CAMP as a second messenger, and it looks like epinephrine, okay? So it looks like a totally different type of signaling pathway, even though the signals are sort of equal and opposite. And I wonder if that has a purpose with complexity, to keep the two signals from directly interfering. You want to evaluate each signal independently and then integrate it. And the only other thing is that you get diseases that mess with this system. A lot of diseases will go after G proteins. And you see right here, in fact, cholera and whooping cough go after the GS alpha exactly. They have an enzyme that forms a covalent bond, and they'll actually take NAD, and it will form this covalent bond to the G protein. Turns out that whatever this is, covalent, very hard to break, and this results in the symptoms of cholera. This results in a misregulation of salt in the intestine. Covalent bond is the real tricky part right here, and the fact is that it's going after this G protein, which shows that it's really important. End result, overproduction of CAMP, and that results in the salt imbalance. So you have a second messenger being messed with by the cholera toxin. The very last thing I want to show in this part, Gproteincoupled receptors are modular, right? That's one of our 5 things. Well, you can see this modularity in the DIY sort of biohacking type movement. Because yeast have these receptors, you can switch out a G protein coupled receptor and keep all the rest of the machinery, or maybe tinker with it a little bit, but it's modular. You can switch stuff out and in. And so what they did is they actually messed with it, and they made yeast into a biosensor. This is how they worked, and the main thing is, don't worry about the details. Just notice that you can recognize a G protein coupled receptor right here. And here's all the stuff we've been talking about. So you can flip in something with a binding site for what you want to look for, the pathogen you want to look for. It'll activate the yeast. It'll change the yeast color. And that's basically what you have to do. Because it's modular, you might have to tweak a few more things. You put the yeast on a dipstick. The dipstick, you dip it into the serum or whatever you're trying to test for. It might be, I don't know what fluid that is, but it might be urine, it might be serum. But it can bind the pathogen. It will produce a color because the yeast will be signaling. And yeast can be dry for a really long time, so you don't have to refrigerate it. Really good biosensor right there. You're mostly just tinkering with yeast, and you're only changing a few proteins. Very easy to do. Because it looks like evolution did this. You know, how do you think we got all those different G protein coupled receptors? Evolution was a modular tinkerer, and so now we're able to follow in those footsteps. Okay, so this last part is actually pretty short, but I do want to separate it because it's